Okay, so we're moving on to animal form and function. So when we look at animal form and function, we are looking at animal anatomy and physiology. When you look at an animal's physical structure, you're looking at an animal's anatomy. The physiology is when you look at how those physical structures function. Okay, so for example, looking at if we take the African elephant here as an example, looking at the elephant's large ears, which are loaded with blood vessels, that would be looking at the anatomy. Looking at the physiology would be looking at how the elephants shunt blood to the surface of those ears uh, to help cool off, right? Because this is what those African elephants do. They use their large ears, they pump blood to the surface of the large ears, and that allows the heat to escape. And if you compare that to an elephant that lives in a cooler environment, like the Indian elephant, you can see that their ears are a lot smaller. So biologists that study an animal's anatomy and physiology are studying an animal's adaptations or animals' adaptations. And then they're looking at heritable traits. So these are what make individuals more likely to survive and reproduce in an environment better than animals that lack those traits. And these adaptations are the results of evolution by natural selection. Adaptations increase fitness, but they're limited by which alleles are present in a population. Okay. However, there is a trade-off in these adaptations, okay, because animals need energy to reproduce. They also need energy to uh, mount an immune response, so they need energy for their immune system. And in many cases, they can't do both, so there has to be some kind of trade-off. So if we look at this of these animals, for example, the decorated crickets, so when they mate, the male cricket will produce a reproductive structure called the spermatophore. So this contains the sperm packet and also a gelatinous mass. So when they're mating, it'll deposit that spermatophore on the female genital opening. Then afterwards, the female will consume the gelatinous mass and leave the sperm packet behind at the genital opening. So as the female consumes the gelatinous mass, the sperm make their way through her reproductive tract. And so the larger the mass or the larger the spermatophore, the longer it's going to take the female to consume it, and the more sperm can fertilize the eggs. Okay? So that's how the cricket reproduces. So what they looked at was if it affected the cricket's immune response, uh, if the energy dedicated to the reproductive output affected the immune system of the cricket or of the crickets. They looked at more than one. So for these experimental setups, they took two groups, so one control and one where they removed the spermatophore from the crickets for 5 days. Okay. Then afterwards, they withdrew the hemolymph from the crickets. So remember, hemolymph is just it's the blood equivalent for insects. And then they looked at um, how much lytic activity was in the groups because they added bacteria to those samples. So after adding the bacteria to the samples, they noticed that there was more lytic activity in the group that had the spermatophores, or there was lower lytic activity in the groups that had their spermatophores removed. Okay? Because lytic activity is just it's the process of bacterial cells lysing, so ripping. That's when like the cell membrane will rip. So that's what they were measuring. And then another experiment that they conducted was injecting the two groups of cricket with either a lipopolysaccharide or a placebo. The lipopolysaccharide was used to simulate a bacterial infection because the LPS is a component of the bacterial cell wall. And so they injected the two groups, well, they injected one of the groups with the LPS and the other one with the placebo. And then they removed the spermatophores to measure the size of the gelatinous mass. And they found that in the group where they injected the crickets with the LPS, the gelatinous mass was significantly smaller than the group where they injected the crickets with a placebo. Okay. So here you can see examples of trade-offs between reproduction and immune response or immune function. An adaptation is a genetic change in a population that is a response to natural selection that's ex exerted by the environment. And that's different than acclimatization, because this acclimatization is a phenotypic change that occurs at the individual level. Adaptations are occurring at a population level. Acclimatization occurs at an individual level. Okay, where an individual will respond to environmental changes. Okay. So these are short term and they can be reversed. And it is similar to acclimation, which refers to changes that occur in an organism in the lab. Okay. So, for example, acclimatization, you can see that in when you uh, when you go to a place with high elevation, so like the mountains in Tibet, or even other types of mountains like uh, like in the Colorado mountains. So when you go to a high elevation, there's less oxygen for you to breathe. So the way that your body acclimates is by producing more red blood cells to compensate for the lower oxygen. And but so that would be acclimatization, so something occurring at an individual level. But when you look at an adaptation, so changes that occur at a population level, if you look at the people that have lived in Tibet for many generations, they are adapted to the environment through genetic changes. Okay. So like the Sherpas that live in like the Tibetan mountains, they they have 5 genes that are identified to help the Tibetans live at high elevations. And some of those code for proteins that assist the hemoglobin in binding oxygen. So an adaptive structure will have its size, shape, or composition correlate with its function. Okay, so because now we're looking at how the structure correlates with function. And you can see that when you look at uh, beak sizes in birds, like the birds that from the Galapagos Islands, you can see that the beak size correlates with the diet that they have. And the way that this happens is through mutations. So mutations in alleles will alter the size and shape of the structure and make it more efficient. And then individuals with that allele will produce more offspring. And then the allele increases in frequency over time in those populations. So here are those Galapagos finches. And you can see that the birds with wider beaks, the more robust beaks, are the ones that are feeding on the larger seeds, and the ones with more narrow beaks are feeding on smaller seeds. Okay, so the form in this case is correlating with the function. But you can also see correlations between form and function at a molecular level, like protein shapes. So enzymes are specific, have a specific form for specific substrates. They don't work with any substrate. You can also see that, you can see this at a cellular level where cells secrete digestive enzymes uh, contain a lot of rough ER and Golgi. And also you can see the shape and function correlate with things like with cells. So the histology lab, you guys looked at simple columnar epithelium. So these are types of tissues that are found in the small intestine. So these are absorptive cells. Okay, and you can see that these cells have a large surface area. Okay, so we'll pause here and then we'll continue with the next video and we'll go into the different types of tissues.